Well, good evening. We're going to continue to look at the embryonic development, and we're going to look at the genetic transplantation experiments. And these experiments, what they found is that there was a real strong link between gene expression and the normal development of the embryo. And as we know, gene expression is what also dictates the proteins. So the proteins were really key for this too. So we're going to be taking a look at these. Now, some of them are very classic experiments. In the 1920s and 30s, and even into the 40s, we had Spemann and Mangold, who were two researchers. Spemann first, and then his researcher, Mangold, she continued the experiments. And they first they first noticed the induction, and these were regions of embryonic tissue that were capable of inducing development. Certain regions. Now, one of the first experiments they did, and they worked on different invertebrates, frogs, salamanders, and in this case, they looked at the embryo number one, and they took a species that had pigmented cells. So they could tell which cells belong to which embryo. And they transplanted this blastopore region, this dark brown region, into the opposite end of this other embryo, embryo number two, and you can see that right here. Now, what they found is, and if you look at a cross section, this is the developing nerve cord. And what they noticed is when they transplanted this blastopore, and this blastopore region into this area, what it did is it developed a second axis. And what they concluded was this is the region that helps establish the anterior posterior axis. In other words, the head to tail axis. And once that's developed, then you can establish the neural tube. And so that's what they noticed. They could actually get one embryo conjoined twins. And they did this in different areas. For instance, this one is the anterior, and this caused a different head region and the anterior portion of the body. And they could also do the back region, and they could find an induced trunk and tail. Now, they weren't aware of what was causing that, but they certainly were the first to kind of show that the tissues could induce development. Another experiment involved the formation of the limb bud. And the limb bud, as you can imagine, you want these cells on the outside of the limb to continue growing. And as they continue growing, you want them to develop into different parts of the limb, the different fingers, the different bones. And they discovered this region in the red that was kind of a control region. And it induced the correct positioning of the limb bud. And so here's another transplantation experiment where they took this red region from one embryo and they transplanted it into embryo number two. And what they noticed is that the resultant embryo developed basically mirror image arms, wings, and they were on the same arm. You can see here's one right there, and then here would be the other one. And that you can imagine what was happening is this one is inducing this direction, this one's inducing that direction. So that was another experiment that confirmed that. Now, over the years, we've been able to refine the experiments. The more we've understood, the more that they were able to then nail down what was happening, down to molecules and genes. We can use transcription factors, and we can even replace the Hox genes that are causing that. So, for instance, in this one that we looked at before in the embryonic induction, we can now see what's happening in that the embryo has these mRNA molecules, hunchback and caudal, and those mRNA molecules end up being transcription factors. as well as bicoid. And so here would be our embryo. We looked at this before, but now we can kind of put it into perspective. The bicoid and nanos, they block. So the bicoid block the caudal translation, and then the nanos is a repressor protein. It blocks the translation of the hunchback. And you can see that the protein density is different. Now, that is one example. And then we also talked about the Hox genes, where we can replace a Hox gene from a mouse and put that into a fruit fly that has a mutation, and we can actually cause the fruit fly to now develop an eye, its own fruit fly eye, not a mouse eye. And so these newer experiments use our newer understanding. And then the other topic that we want to cover is another molecule that is really important, not a protein, but a microRNA. And a microRNA is a short strand of non-coding, and non-coding in this case, remember, RNA we think of usually is going to be translated into a protein. Well, these are non-coding RNA molecules, about 22 nucleotides. They're very short, and they're important because they can bind to specific mRNA. So these are microRNA, and they're going to bind to mRNA. So let's take a look at this nice picture here. Here would be a microRNA gene, and it's going to be transcribed. And up here on the top is a typical gene. Now, a typical gene, we're going to transcribe into a messenger RNA, and then that's going to be translated to a specific protein. But what if we don't want that protein to be made? Well, in the developing embryo, we want to tightly control what protein is made where and when. And so these microRNAs, they can bind to it. So just a nice model here. Here would be our mRNA. And we can just, put down some letters here, A, U, C, C, G, A. And the microRNA would bind to it, UAGGCU. And because we know that these base pair, we form hydrogen bonds, we would see that binding occur, and now that protein assembly is blocked, that translation is blocked. So microRNAs are very, very key in the embryonic development, and we see these being produced in different parts of the embryo. Here's one experiment where they were looking at a normal embryo, and we call that wild type, and you can see down here in the bottom, this is showing the resultant proteins that are being made in orange. If we remove the microRNAs, if there's a mutation, or we block the microRNAs, notice we don't see those regions of orange anymore. That shows a difference in the proteins that have accumulated. And if we add back that microRNA, now we get the regular development. And if we add back another molecule that blocks the microRNAs, we get our experimental results again. So we see that, and we are seeing that more and more, the microRNAs are involved. And then the last bit we need to cover is the programmed cell death, apoptosis. Now, apoptosis can happen in multiple areas in adults too. Trees even will show this. In the fall, the part where the stem of the leaf, the petiole, is attached to the branch, that will slowly die, this region, and then that's what causes the leaves to die, and then they will drop off. Now, programmed cell death, in the embryo, these are going to be seen in places where we want to control where cells are going, or in this case, not going. So notice in this picture, we're going to see the first example is when we want to remove tissue, and the example here is in the metamorphosis of a tadpole to a frog. We want to resorb that tail tissue, and then those materials, those resources are then used to build and sculpt the adult frog. In other vertebrates, We see this in humans as well. We want regions that correspond to the fingers and the toes. In between the fingers and toes, we want these cells to die. So notice the black arrow here. These are all cells that are slowly dying. And as we are growing our hands and feet, these areas, as they die, will allow us to have a separate fingers and toes. So the apoptotic cell is being told, getting signals to die. Now, in all of these, you can imagine that any mutation involved would cause abnormal development. So we see mutations in the transcription factors, and the transcription factors, again, if they have a specific shape that is supposed to join to a specific DNA sequence, if that shape is different, it's not going to be able to join to the DNA. Mutations in receptors, if you have a specific receptor on a cell surface, if that receptor changes shape, you can again imagine that receptor no longer binds to those growth factors. And then you can have mutations and timing events. When are they made? What happens? Even in cilia help move cells. So if the cilia have mutations, we're going to have problems in telling cells to actually move. So you get a glimpse into how important these are. These genes can even be involved in adult problems. A lot of these growth factors, a lot of these genes, if they have mutations in the somatic cells, they can cause cancers. That's become a real hotbed of research there. Well, I hope this helps you understand more about the embryonic development. Thank you for watching.